All right, so why don't we kick off? Welcome back everyone to our first snack of 2022. It's my pleasure to invite Henning Sprekeler, and Henning's work has been some work that I've really enjoyed over the years. It's really fun to kind of get to meet someone whose work you've read for a while, at least maybe not in person, but at least face to face on a screen. And I first came across Henning's work when I was trying to think a little bit about burst firing dynamics and layer 5 pyramidal neurons that we've thought about a lot together as a group, and in this snack, thinking about Matthew Larkham's work, but also a lot more recent work from a number of groups. And Henning had this really lovely paper that was thinking about the difference between spiking and bursting in pyramidal neurons and whether or not they could convey different messages in multiplexing, which I gather he might get to in the second part of his talk. And then just recently, I saw a really lovely bioRxiv paper that was sort of gesturing more towards the sort of realm of deep learning and thinking about how these different kinds of multi-compartment models of neurons might convey particular capacities to the sort of systems level stability of a network. And I thought it was a really lovely piece, and it actually meshes in quite nicely with the perspective that we wrote on the sort of neuromodulatory system and its effects on plasticity and burst firing dynamics in the brain, just recently with Michael Breakspear. So I'm really excited to see what you have to say today, Henning. And thanks again for joining us. Take it away.
Yeah, thanks a lot. Thanks a lot for the introduction. Thanks a lot for the invitation. And thanks a lot to all of you for being so flexible with respect to timing. We'll see. I'll have my coffee, you'll have your beer, and we'll take it to hopefully a nice place. As Mac already said, please do feel free to interrupt me at any moment in time. I think discussion, if I should lose you at some point, if there's an interesting point on the way, if that gets lost, it would just be a shame, right? So, so the talk is basically two parts, right? The first one is more about this recent bioRxiv paper that Mac mentioned, where it's sort of about invariant processing. And the other one is a little bit more low level, if you wish, about sort of dendrites and what they could do. This first part in particular is really not work that I've done, but work that was done by these two super talented PhD students who I'm very happy to have in the lab and very unhappy to lose very soon. So they'll basically, they're the ones who did the work, right? Okay, so all of you are familiar with this idea, I presume, of hierarchical sensory processing, right? So it's this idea that you basically have sensory data. Can you see my mouse? Cool. So there's basically sensory data that are coming in from different sensory modalities, and they're processed through, maybe thalamus, earlier cortical areas, higher cortical areas, etc. Maybe for audition, there's a little more sort of low-level areas in between. But the overall motif in which people think about that is that just information is relayed, and on the way, more and more complicated features are extracted. Right? That's sort of this sort of deep learning view of the brain, if you wish. Right, and that's also something that people like Jim DiCarlo have picked up recently. They've really exploited that to see how far can you take that deep learning or deep network allegory to explain data in cortex. Now, in vision in particular, there's one function that has always been conjured up, which is also maybe also the thing where the breakthrough of deep nets have really taken place, which is object recognition. Right? And object recognition has that problem of requiring invariant object recognition, because if you see an object, you never see that again in exactly the same configuration. You need to see it basically maybe in different lighting conditions, different angles, different sizes, etc. So if you want to recognize an object, you invariably have the problem of overcoming these contextual variables that sort of pollute the low-level representation of that object. Right? And sort of one of the ideas is that that's what's basically happening along that hierarchy. That you might have low-level features where if you look at the representation of objects, basically in that high-dimensional pixel space, maybe, then this representation looks really sort of, yucky, right? It's very hard to understand and see what's going on. Whereas if you move to higher-level areas, you eventually get to a situation where you can linearly separate, different objects from each other, but basically just a linear classifier, right? So that's sort of the idea. And so that's all the view of feed-forward processing. Right? Now, that has changed a little recently because we know that there's really not only feed-forward connections in the brain. There's an abundance of connections that go backwards. And that is true in all of these sensory hierarchies that I've shown you here. That is true even if that people have known that forever, right? So if you look at the sort of famous Velleman van Essen figures, it's in there. Gilbert has basically spent his whole career just characterizing these types of feedback connections in visual processing. There's a lot of stuff going on there, and I think we have a lot less sort of theoretical grounding or sort of idea of how to understand what these things do, specifically on the neural level. Okay. So if you think about those sorts of things, then of course there's a lot of computation that people have conjured up for feedback connections, right? So there are these sort of classical things like predictive coding or sort of generative models for the statistics of the data. There could be something like valence signals, right, that allow you to learn something, or error signals that also allow you to learn something, or other things like attention, contextual modulation, inference copies if you actually have an embodied agent. All of these things have basically been implied or implicated in feedback signals. But the thing that is a little bit hard is that from that level, getting down to the level of neural recordings is not necessarily trivial. Okay, because that's the point where we can do amazing stuff there as well, right? We might have viral tracing and find out where the signals come from. We can image axons, recently even boutons, right? We have this population level activity where we get more and more neurons that we can record at the same time. But it's a little weird because we have this computational framework and we have that implementation level, and mapping those two is not always trivial, right? And so that's a little bit what I'm trying to talk about in the coming few slides. And so the approach that we've taken basically is if you wish, an optimization or deep learning approach. Okay? So we've basically taken a network that looks like a biological network that has feed-forward elements, that has feedback elements, that is, has excitatory and inhibitory neurons, all sorts of stuff. That allows us to look at it as if we would look at, a little like we would look at neural data, right? On the other hand, we want that network to do one of these things here, or want a specific function. So we go ahead and we train it using basically machine learning tools like autograd, gradient descent, to perform the function that we want to instill into the network. And then afterwards, we can analyze it and see how it solves it. Right? Sort of that approach that a lot of people have adopted recently to sort of try to see how more complicated tasks could be solved on the neural level. Okay. So let's jump in. The type of task that we concentrated on is blind source separation. Okay, so for those of you who are not familiar with it, it's very intuitive. If you're in a room and a lot of people are talking at the same time, then of course there is this wish to maybe concentrate on one specific person at a given moment in time, right? So it's sort of like suppress the blur of all the other people. And that task is many decades old. It's something because sound waves superpose linearly. It's a linear separation task, basically, that you have to do if you want to linearly mix things into separated items. And that is old. It's basically solved by independent component analysis. Okay, so that's the rough setting. So you have sources. They vary in time. They give rise to stimuli, but these stimuli are a mixture of the original sources because you might be listening to several people at the same time. And then from that, you basically want to recover the original sources. Okay, so that's the setup. And the little twist that we've added to make this less of a trivial task is to basically say that the sources, or the way the sources are mixed, is something that changes over time. Right? So the allegory is basically that if you're in a blind source separation setting or a cocktail party setting, that people are moving around in the room, right? And that you basically have different mixtures over time that you need to unmix, right? And at that point, it's no longer a simple ICA problem, but it's actually a collection of ICA problems, and you need to adapt at any moment in time to find out how you should solve the task. Okay. Now I'll talk a bit about it in a second. You can think about this task in many different ways. You can think of it as an attentional task where you need to dynamically, address your attention to certain places. You can also think of it as a meta-learning task, right? Because every individual blind source separation task can be thought of as one that you need to learn. And so the whole collection of them together is a meta-learning task. So for the more deep learning aficionados out there, right? So there's a lot of different ways of looking at this problem. Okay, so just to get the language right, there are the sources, there are the stimuli, which is basically this mixture. There's the output that you try to infer, and the hope is that this output is basically invariant to the mixture, right? And the mixture is changing over time, and I'll call that context. right? Just to stick to the notion that I had earlier for the visual situation. They're just contextual changes, and there are changes in the sources themselves. Okay?
So how would
so you
that
have
be unpacked, Henning, from the idea that you could see the same set of inputs but have been told to respond to them differently? Like, you know, let's say you trade me up to do a go, no go task where green equals go and red equals stop, and then you switch me and say, now all of a sudden red means go and green means stop. And so I'm getting the same input, but the rule you've given me determines how I should respond. Would you have the flexibility to model something like that? Or are you just interested in the context being the complex signal that's coming in at the start, so the sort of configuration?
That type of problem is inherent here. Okay, because this is, you can, and that's actually the reason why you can't solve this task in a feed-forward manner, right? And now, so the reason is that basically the sources, if you wish, are they live in a two-dimensional space, right? So if you change the context, they're mixed in a different way. So that means what was previous, you know, the semantics of one specific data point change over time. In a different context, they need to be processed differently in order to achieve this invariance. Right. So that's, in that sense, this is, you can think of it as a context invariant task, in the sense that you need to, you know, ignore the context. You can also think of it as a context dependent task, in the sense that the same stimulus needs to evoke different responses depending on the context that you have.
Okay.
Did that clarify the question?
Yeah, that's great. Thank you.
And that's actually the reason why you need feedback for this, right? So if you don't have these sorts of ambiguities in the system, you can solve it feed forward. Right? But as soon as you have these ambiguity, you need to do a dynamic inference of which context is actually present. Right? And the task that you mentioned earlier, this sort of green, red, or, you know, motion versus color, these sorts of Valerio-Mante tasks, that it's the question: do you tell people that you've changed it, right? Or does the network or the animal need to do a dynamic inference of what's going on in the style of like a Wisconsin card sorting task, right? So that's I think the way I think about it. And here you really need to do the dynamic inference, right? So you don't know, you're not told what the context is right now. You need to find out yourself. Okay. Good. So and the way we solve this is, as I said, through feedback. And the idea is that there's some kind of modulator that could be located anywhere, somewhere in the brain, right? That basically just sends feedback signals and changes an inherently feed-forward pipeline of sensory processing. Okay? So that's sort of the view that we adopt. And the reason why we adopt it here is that the task itself in any given context is still a linear task, right? So of course you could just throw like a complicated LSTM at this, or a complicated recurrent neural network to solve the task, but that ignores the fact that in every context itself, it's a linear problem. Right. So in that sense, that's why basically we adopted this notion that you have a linear network, but there's just something on top that changes it according to contextual demands. Okay. And I think this idea is one that could be interesting to follow up in several ways. Good. And that modulator obviously needs information, right? So it'll basically receive information about the stimuli that the network is receiving, or that the brain is receiving, and, you know, about the output, right? What is it currently doing, right? And so those two things should then allow it to infer what the context is and go ahead and you know, revert it if you wish. Good. Okay. So let's see if that works or how that works, right? As I said, we're just going to optimize this RNN. Maybe a notion, because that's one thing that the reviewers didn't like so much, is the fact that we're not going to dig very much into how the modulator does this. Okay, we're just going to optimize it, and then afterwards look at what's going on in this network, because that's probably where we can pinpoint better where we're going to look, right? If we do an auditory task, then you'd look at auditory cortex. Where exactly that top-down stuff comes from is a lot less clear. That's one aspect. That's just sort of the utilitarian aspect of biology. The other aspect is, of course, that what exactly the modulator will do will depend on the problem at hand, because it's a dynamic inference task. So depending on which statistics you're going to use for the stimuli and which ensemble of contexts you're going to use, that modulator will do different computations. So it's a little bit idiosyncratic what it does. And so that's why I think it makes limited sense to like go in depth. Okay, good. Let's dig in. So what I'm going to show you here is now these different signals that you have in this hierarchy: the sources, the mixture, and the unmixed things, right, in two different contexts. And here's basically the whole thing unfolding in time. These are the signals. These are the two mixtures. These are the outputs. And you might see that there's these dashes on here. It's a superposition of the sources and the thing that is extracted, right? And the fact that these things basically lie perfectly on top of each other means that the network is solving the task. Okay. Now you go ahead and you change the context abruptly at one moment in time. And what you see here is that very quickly, the network still does the task fine. And that's just a tiny little bit of a deviation here at the beginning. That's the time that the network needs to find out what the new context is. Okay. And if you quantify that, then there's a deviation in the accuracy of the unmixing that occurs very briefly. There's a little blip here in the error that the network performs. Okay. So if you look at this, what this modulated does, it really switches over time in a step-like way for different contexts. Okay, so that means it really does basically an inference of the context. It doesn't, invent the signals or do something like that. It goes ahead and really just changes what the configuration of the network is for a given context and switches between those. Okay, a little bit of quantification, right? We have the stimuli, we have the output, and obviously you can go ahead and basically calculate correlations here, right? So that just basically to quantify how well the network solves the task. Obviously the stimuli are just a mixture of the sources. So if you calculate those correlations, it's going to be whatever, right? And if you look at the output, then the ideal situation is that, of course, the output 1 is perfectly correlated with source 1, output 2 is perfectly correlated with source 2, and there are no cross signals in here, right? And so that basically allows us to calculate something like signal clarity, which is just the mean absolute difference of the absolute correlations. If that's 1, the task is solved perfectly. If it's 0.5, it's random. Okay, so that's just as a quantity that you'll see a little over the time. Okay, so far it's all good, but this is a very abstract network, right? It's all linear, it's an RNN, it's hard to map it to the brain. So what we're doing now is basically to gradually make it more and more biologically plausible, right? And so the first step is to think about time, right? To think about feedback signals potentially being slower in their action, right? And that would certainly be true, or likely be true, for classical neuromodulator, be it acetylcholine, dopamine, or whatever, right? Things that I think you've thought about quite a bit, Mac, right? And then there's also this sort of say cortical-thalamic loop stuff, Murray Sherman, so who've basically also invoked these connections to be more metabotropic, right? Which is also a slower signal. So we need to think about whether time really matters here. So we'll vary the temporal timescale of the feedback. And the second thing is then to vary the spatial precision of that feedback, right? So is it really necessary that this is something that, modulates individual synapses, which would be, highly unlikely that this is really done? Or is this something that can be a very coarse signal? Right? And so the first point on slow modulation is actually very easy to implement. What you do is you just basically, you stack a low pass filter on the action of the feedback, right? You just suppress high frequency signals and force it to be a slow thing. And then you can check, how much that damages the performance of the system. Okay. And so if you plot the signal clarity as a function of, how much you suppress high frequency content, then you see that, if it's very fast, it's almost perfect accuracy. And then as you gradually increase the time scale of the feedback signals, you get a very graceful and graded decline in the performance. Okay. And so this, what you see here on the right is something that is actually very slow already, because the time scale that matters are the contextual changes, right? So if something changes on a time scale of 10 seconds, obviously if your feedback is acting on a time scale of an hour, you could just forget about it, right? So basically 1 here is the time scale on which the contextual changes happen, right? So the take home here is, as long as you're mediocrely faster than the contextual changes, you'll be doing fine. So yeah, that's the readout, which is now basically, if you wish, the way the modulation looks. And I'd like to address your attention a little bit to 2 things. The first one is, if you make this slower, you're going to see that there are longer transients, right? Which is to be expected, right? Because the signal is slower. But there's also less noise. Right? So what you see here is a classical sort of speed accuracy trade-off, if you wish, right? If that feedback thing is slower, it has more time to integrate information, and as a consequence, it's more accurate, right? On the other hand, it also needs more time to get it right, right? So that basically means that you're basically going to see something, if you look at how well the system does it. If you're super slow, you're bad, right? If you're very fast, you're getting it very quickly, but you also, plateau on a comparatively high level. And if there's an intermediate time scale, then you're actually having the best performance. Okay, which is sort
So
of
Henning, if the feedback's too fast, can you have the network make false positives? So if you have a noisy signal coming in, can they think that it was one when it wasn't, when the evidence was maybe sort of a bit more ambiguous? If it's really fast, but then you imagine if it's slower, there'd be more time to kind of arbitrate over it. If the speed accuracy trade-off analogy is good, then you'd be okay. Oh, originally I thought it was one, but no, that was just me guessing a little bit too early. Now over time, it's become very clear that it was two, just a noisy two. Can you find things like that?
I would imagine that you could, but there are two things that come in, because as you say, if you inject noise, just noise, then you'll see exactly that sort of thing. If you go ahead and you give something that really looks a little different, right, then it's the question how well the network generalizes, and that's sort of yet another question where I could imagine that you could get all sorts of artifacts, because it is something that has learned the statistics of the data. Right? It has an internal model, if you wish, and if you give it something for which that model is inaccurate, all sorts of stuff is going to happen. Right? But otherwise, yes, that's kind of what I would expect, that this is more pronounced even in the presence of noise. Okay, good. So briefly, what input does the modulator need? So we can now do all the things that are hard to do in the brain, right? We can basically lesion connections. We can lesion feedback connections and look how well the system still works. And as a function of time, it performs okay for a while, and then it breaks off quite drastically. Okay, so performance is impaired if you remove top-down connections, if you wish. If you remove feed-forward connections, the effect is even more drastic. Okay, because the system doesn't know what the mixture is. Right? So, in that sense, you basically need to you kind of need to combine both, at least when things are slow. It's still okay if you only have feed-forward. That's something that is, I think, a little bit inherent to the problem. We could talk about that in detail at a different point. Okay, spatial imprecision is the other aspect, right? So are the neuromodulators something that is really a very precise action or not? I think that's not resolved. We don't really know. But there are these aspects of feedback arriving in layer one, and then in layer one there might be neurons that actually do volume release of GABA, like NDNF neurons. There's all sorts of stuff sitting up there that actually is comparatively diffuse in action, right? So in that sense, we wondered whether that is a deal breaker, and it turns out not really, right? So the way we looked at that is to basically say, hey, we now go more to a neural representation. We take our mixed signals, which would be sort of our raw sensory data, and we embed them in a population of neurons that basically now has random tuning to these stimuli. And then afterwards, we ask if we can decode from that population the original sources. Okay, so that's sort of the setup now. And what that allows us to do is to range all these neurons in a spatial manner, and then afterwards provide feedback that is spatially diffuse. Right? We need a notion of space, otherwise we can't talk about diffuse feedback. And so now the feedback goes in and acts on neighboring neurons in a similar way. Right? So if you wish, it's a kernel, so you have a certain number of feedback signals that go in, and these different signals target different places in that spatial organization. And we can now control, one, how many feedback signals do I have? So what's the dimensionality of the feedback? And the second thing we can control is sort of how precise it is by changing the width of action of these different types of feedback by just plugging a Gaussian kernel on it. Okay, so that's the idea. Now we can go ahead and study how well things perform. And so what you probably see here now is that as a function of time, you see the error, and there's still the same thing. There are short blips after a contextual change in the error, but it's very quickly healed. So the network still does the task. And this here is basically the modulation factor that is acting on the different neurons. And you see that this is now a diffuse thing across the 100 neurons that I've embedded here. And you see that there's sort of blob of modulation that is traveling in time, right? Which just changes abruptly at the moment when the context changes. And so if I now plot just performance, the signal clarity as a function of the number of feedback signals, which is fine for just 4 feedback signals. In fact, 3 are enough, but that's we can talk about that otherwise. And the modulation width is something that can be quite broad, right? Because you can just, well, in the end, the task itself is comparatively low dimensional. If we go down that line, right, it's a 2-dimensional input, it's a two-dimensional contextual space. So in the end, we're living in a very low dimensional problem here. So it's not very surprising that you could do things with 4 signals. Okay, if you would go to something more complicated, you probably need more. Right? But the diffusivity itself is not necessarily a deal breaker as long as you're like sufficiently complete. Okay. Good. Now that's just diffusion. And now let's think a little bit about how to basically look at that on the population level. Right? So things that people would record might be, via calcium imaging or whatever, neuropixels or whatever your favorite sport is right now for electrophysiology. You can try to think about, what kind of data would I acquire? And to do that, we basically extend the network by one additional step. And we say that there's like a low level sensory area, right, which is one that is not going to be invariant to context, okay? Because we just project sensory data into that area in sort of a fixed static way, right? It's not subject to modulation. And then we have a higher level sensory area where we assume that modulation is acting. Right. So of course, it's a bit of an artificial dichotomy, but it might allow us to contrast what should happen in low level and higher level sensory areas. And then, of course, the task is to decode from a higher level sensory areas afterwards the signals that were originally going in. Okay? So the same setting. And now we just go in, we train that thing, of course, again with the modulator. Right? The modulator acts in a diffuse way on that higher level area. I think it's also a little bit slower. Actually, as I said, it's actually beneficial to train things. And then afterwards, we can go ahead and think about how it acts, right? And so the way we did it here is because A, we have a little bit of a crush on presynaptic inhibition, but B, there's just this idea that this feedback stuff just provides a gain modulation, if you wish, of the responses in these brain areas. And so you could simply say, hey, my neurons, individual neurons are just, they ramp up in gain or ramp up or down in gain, and that's all that we do. Right? So we don't modulate synaptic connections or something like that. We do a comparatively coarse thing that just acts on the gain of neurons. Good. So let's see what that does. So basically we have the output at the higher level, which is just the sum of the inputs from the lower level, which is now excitatory input, right? And then there's something like inhibitory input that we have at the same time, right? So that we can distinguish excitation and inhibition, which is also our personal crush in the lab, right? We love excitation and inhibition, specifically inhibition. And then at the beginning, you basically have that gain modulation factor that changes how that specific neuron is going to react. And that is specific to every neuron, albeit diffuse, right? So neighboring neurons are going to have very similar gain modulation. Okay, good. I think I'll skip over those things. There are things like excitation and inhibition actually being correlated, but I think I'll talk a little bit more about sort of the inner workings. Okay, so what do we see? Same thing. Gain modulation is something that is now changing over time. Gain modulation is now a factor between 0 and 1, meaning if there's no feedback, then the neuron is not suppressed. If feedback comes in, it is to some extent suppressed in gain. Okay? This is sort of the idea that this is an inhibitory action coming from layer 1. And the error is the same thing, right? There are just blips after contextual changes, but overall the network still does it. Now, the thing that's maybe more interesting now is to look at what the responses of these neurons actually look like, okay? And so now this is now, if you wish, something like a raster plot or so, right? I'm just plotting activity of the 100 neurons as a function of time for two different contexts, right? And this is now the higher level area, not visual, so it's just an area. And so I think what you can tell already here is that the responses in one context are different from the responses in another context. Okay, so it means on the level of individual neurons, you don't achieve a context invariance, right? But you must achieve it on some other level, right? And that level turns out to be the population level. And I'll talk about that in a little bit more detail in a second. So, for example, if you look at this here, then I'm basically just plotting the firing rate of the collection of neurons in one context versus their firing rate in a different context, and you see that these things are not clustered around the diagonal. Right? So that means the firing rate of these neurons really changes from context to context. Although in the end, that's the place where the modulation happens and where the invariance is achieved. Okay? So it means the invariance is not happening on the single neuron level. Okay, so how does that work then? If we look at individual neurons, let's just flesh it out a little bit more. We could look at what the signal clarity is in individual neurons in the population. And it turns out that that's pretty much close to chance. Okay? So it's basically about 0.5. So this is pretty much arbitrary mixture. If you look at the clarity in the higher level visual area, it's the same thing. It's pretty poor as well. Right? If you look at the modulatory fibers that come back in and you try to understand whether they represent the signals, that really sucks, right? Which is not surprising, right? But if you look at the output, it's perfect. Okay? So that basically means that it's really just on the single neuron level, there's not much to be seen. And now you can flesh that apart even more. It's just correlation for signal 1, correlation for signal 2, and there's a very broad spectrum of correlations with those two things across the neurons. And so the other thing is that, the clarity of, you might suspect that one neuron is very clear across all the contexts and that the decoding just relies on that single neuron. That's also not true. The clarity changes across contexts, right? So if one neuron in one context really just represents one signal, then it represents something completely different in another context. Okay, good. So that's not invariant, but on the population level, it is, okay? And the way we check that is to simply say, hey, we have our high level population, and now we just train a decoder, right? Basically, the way you would do it if you had high dimensional recordings and you knew the sensory data, you could just go ahead and do supervised learning on that linear development, okay? To see how information is there. And so just to sum up briefly, of course, in the mixture, from the mixture, you can't decode. From the low level sensory area, you can't decode either. But for the high level area, you can decode perfectly. And what's important now is, of course, that we use exactly the same decoder for all contexts, right? Because otherwise we'd be cheating. Of course, we could decode for every context individually, but you need one that actually works for all of them in order for this to be a viable check. From the modulatory fibers, you can't decode anything. And then, of course, the output is perfect. Okay. Now, the other thing you can think about is to decode the context, right? And so that's something, because this is inherently what people would probably call like a mixed representation, right? Or sort of what does Stefano Fusi call that? Sort of, it's like a nonlinear expansion of the data, and those data are both the signals and the context. So this is a mixed selectivity. I think that's the word that people use, right? So if you try to decode the context from the lower level, it doesn't work. From the higher level, it works fine. The thing that we found surprising is that you can't decode the context from the feedback, okay? Which is sort of an interesting thing, because you would expect that's where the context information is, but you can't decode it. And now what we figured afterwards is that this is something that simply has to do with linearity of representation, right? So the information about the context is perfectly present in the feedback connection or in the feedback projections, right? But you can't decode it with a linear decoder. And the reason for that is very simple, namely that the representation is so nonlinear that a linear decoder sucks. So this is a depiction of the different contexts we have. Okay? I'm not going to go very much into detail here. Just sort of think of the colors that you can think of these as two triangles with sort of bright colors at the outer edges of those triangles. Okay? Now, if I go ahead and I plot the responses of the feedback connections after basically doing PCA, what I'm going to see is that these triangles are also there. It's just that they're flipped. They're flipped inside out. So those points that are here are at the center here. And this point here is at the center there. And the reason is simply that what we're putting in as a context is a mixing matrix. And what we need to do is to invert that mixing matrix, right? So there's an inversion operation in the game. And that inversion operation simply means that, an inverse is not very well decoded by a linear decoder. Okay, so if we decode the inverse of the mixing matrix, we do perfectly. Okay, so which I think might be an interesting sort of just take home when we think about contextual signals or feedback signals and thinking about those as contextual signals, because even if we don't manage to decode from them, it doesn't mean that the information is not there, right? We should be careful about that. Okay, good. So I think the last thing I have is a little bit of eye candy. And then we can talk about something else. So the Eyecandy is now the idea of visualizing what's going on here, what the feedback does at the level of the population in sort of a three-dimensional video. Okay? And just to be clear, I'm not good at Eyecandy. Laura is, who is the first author here, right? She's a magician on that front. Okay, so as before, we basically have the source and the unmixed source. We have the second source and the second unmixed source as a function of time, and this is sort of the modulation that's happening. And what I'm plotting here on the right is a little bit more complicated because I have that higher level activity in that higher level area, right? Which is, I think, 100 neurons. And for those 100 neurons, I want to visualize that in a three-dimensional space. I have to think about which dimensions to use, right? I need to project it down to 3 dimensions. And the 3 dimensions we're choosing is basically the readout dimensions, okay? Because those tell us how well the system, basically untangles the sources. And the 3rd dimension is just added to show us what happens in that high dimensional space to the representation of the sensory data. Okay, and so for that we chose the first principal component. Okay, now,
I mean, did you calculate that on like a trained network, or did you calculate it sort of as the network was training? How did you decide what to do there?
You just train the network to perform the task, and then afterwards you feed in the signals and you just do PCA.
Right? Great, and so you just take the loadings of the first PC. Cool. Yeah, it makes sense.
Yeah. So in a way, the analyses we've done here, we try to do them such that they would be doable also for somebody who has recordings, right? And specifically these 2 dimensions here, S1 and S2, aren't really the readout dimensions, but they're actually like a trained linear decoder for source 1 and source 2, right? And they might appear subtle, but those things don't necessarily have to be the same, right? Because there could be ambiguity. There could be more than one direction at which you can look at the data and extract the sources, because it's
that actually gets back to Carlo and Cox's idea, right? That you could see a face in multiple different orientations behind a fence and upside down with a hat on, and it's always the same face. So you want to get the concept, the invariant concept, rather than worry about idiosyncrasies in the input. So yeah, I think that's cool.
Yeah, exactly. Right. And so what the other thing we did is we just used as inputs now simple sinusoids, because that makes the visualization easier. Okay, and now if you take 2 sinusoids with 2 different frequencies, you put them onto 2 axes, then you get Lissajous figures, right? So it's this sort of classical figure 8-like shapes that look like that. Okay, and so if everything's doing fine, if the network is performing fine, you should see this gray dot here tracing that green outline. Okay. Now the high dimensional representation of the 3-dimensional projection is going to move in 3D, and we're just going to project it down onto those 2 dimensions down there, which is this gray dot. And if that traces this green curve or green bluish curve, then it does it fine. Okay. Okay, so let me show you how that works just for the beginning. So you're going to see this trajectory moving in that 3-dimensional space. And if you look at the projection down here, it's tracing the outline perfectly. So in that one context, it's actually solving the task well. And you see that it's actually just a 2-dimensional embedding in that high dimensional space, which is not surprising because it's a 2-dimensional signal, right? And if you look at it from the top, then it looks like it's just unmixing everything fine. Okay, so far so good. And now we'll do something weird that is very hard to do in an experiment. So what we're trying to do now is to introduce a contextual change. But a contextual change would change 2 things in the network. It will change how the feed-forward input comes in, right? Because there's a change in contextual representation. And it will also change the feedback that's coming in, because the network is trying to solve the problem by invoking the feedback. And of course, it would be good to see what these 2 things do individually, right? And that's something that's hard to do in experiments, but in a model you can, right? You can just go ahead and freeze one of them and just change one. So this is the first step we're going to do. We're just going to switch to the different context, but we'll freeze the feedback at the level where it was for context 1. Okay, and we're going to check what's happening here. Okay, let's play. Oops. So you see that everything's still moving fine. Now the switch should come any second. And now you see that it's no longer tracing things fine down here. Okay, so the feedback is actually necessary to solve the problem. And the other thing you're going to see is that it hasn't left this 2-dimensional manifold, right? So this is basically a sharing of the representation in the manifold that it had before. Okay. And then at some point, what's going to happen is that we're going to switch on the feedback again. Okay, so we can just release it from the freezing. and let the network do its inference task. And this should be happening any second now. And now, yes, here we go. So now it takes a while to reorient itself, but then it should be fine at doing the task again, okay? And so now, of course, that looks weird at the beginning, but once the trajectory is over, it's fine. And the thing that you should probably see now is that at this point, it has left the manifold that it had before. And so basically, the change in context kept it within the original manifold and sheared it within it, thereby destroying the mapping. And then afterwards, once the feedback comes in, it sort of just rotates things out in order to see it well. Okay? So the way I think of this is that you basically look through a lens and you have something like a Rubik's Cube below, and somebody goes ahead and changes something, and by feedback, you just go ahead and rotate it back so that it looks the way it did before. Okay? Which is actually something that goes back to old ideas like, Bruno Olshausen had this stuff in the early 90s, right? It's sort of this idea of normalization of the sensory data. Okay, good. So just summarize, there's a 2D population manifold or manifold on the population level. A changing context rotates within manifold, and feedback reorients that manifold such that you can afterwards basically look at it the same way from the side of the output. Okay, good. So I think that's pretty much the end of that story. Let me have a brief look at what time says. I think I'm already late, right, Mac? 7:45.
It's all good, Henning. That was really great. I'm very keen to hear a little bit about the multiplexing stuff, and in particular to sort of pass some of that by Joe's filter. So if you have extra time to cover some of that, or even just sort of cover some of the key concepts, that would be awesome for discussion.
Yeah, there would be. That's very easy. So I mean, I think I can try to do that comparatively quickly as well. Then I'll just jump over this summary here so that we don't have to torture you with that. Come on, you can do this. Let's go. Okay, same thing in a sense, because it's still about bottom-up and top-down signals. Okay, so we're switching to a little bit of a different setting now, however, because we're not thinking about bottom-up and top-down signals, if you wish, on the functional level, which I tried to do now to see what the computations are that are performed by top-down and bottom-up, but more on what's actually happening at sort of a much more fine-grained level of neurons, right? Or neural populations, and those are actually spiky neurons. And I'll worry a little bit about the actual electrophysiology of the neurons that are the receivers of feedback signals. So as many of you probably know, at least Mac, you said at least your lab, you thought about these things quite a bit. The neurons that are sitting in cortical areas that are, receive top-down and bottom-up input are classically thought to be pyramidal cells. Right? And pyramidal cells have this sort of beautiful structure where they have their cell body in layer 5 or layer 2, 3 or so, and then they have these really long protrusions, sort of the apical dendrite that projects or that collects information all the way from the very top of cortex from layer 1. Right? And that's sort of the place where the classical feedback projections arrive. They arrive in layer 1 and sort of allowed to process that information in the apical dendrites of the pyramidal cells. And then there's this bottom-up input that is basically arriving through layer 4 and then basically reaches the basal dendrites of these cells. And so there's basically a morphological separation of bottom-up and top-down signals. And so now the question, of course, is how do these neurons combine these two things, right? And so there's a lot of things that one could imagine, right? You could think about this sort of predictions with sensory data in sort of Bayesian Q combination way, or you could think about the one being error signals that allow you to learn the lower stuff, right? There's a lot of ways in which you could think about this functionally. But I think one thing that is important is to think about whether these two signals that are probably like really different in nature, how they're just clumped together. Right? And the classical views, of course, is that there's a single output, it's just the spike train of the neuron. And that output depends on both of these signals, but you project 2D down to 1D, and so there will be information that's lost. Right? And so that's sort of that idea is what we want to question. Right? And so we're going to question it based on how these neurons process information. And that goes back to the work of Matthew Larkham, who's basically shown that if you patch these dendrites up here, that there's a very nonlinear phenomenon that can happen. Right? If you just provide dendritic input that is, well, you basically patch the dendrite, you provide a little bit of input, you're going to see sort of a voltage excursion. If you stimulate the soma alone, you're also going to see a voltage excursion, potentially with a spike at the soma. But if you do both things at the same time, something superlinear happens. Right? And that superlinear thing is basically the fact that the spike that you might trigger at the soma at the beginning back propagates into the dendrite. And then sort of there sums up with this dendritic input that you have provided. And those two things push calcium channels across their activation threshold. And suddenly, you get a rush in of calcium. You get something that people call a plateau potential in this apical dendrite. And that plateau potential then in turn sends a wave of depolarization to the soma where it can trigger additional spikes. Right? And then you get a bit like a ping pong between those two. And in return, you're going to see not a single spike, but actually a sequence of action potentials, meaning a burst. Right? A burst of 2, 3, 4 spikes in very rapid succession. And so now you can basically try to interpret this in different ways. And so the classical notion is, of course, that, from 1 spike to 3 spikes, hey, that's gain modulation, right? You're going to have more spikes than you had before. But if you actually think about the spike patterns themselves, then there's actually more information than that. Okay? And so let me just quickly sort of outline the assumptions that we made. The first one is we're going to assume that it's not a single neuron, but we're going to move to something like an ensemble of neurons that sort of might be part of a cortical column or something like that, neurons that are semantically similar. Okay? And I'll assume that, as Matthew showed, somatic input generates spikes, right? And dendritic input converts spikes into bursts. Right? That's sort of basically what's happening here. Somatic input alone generates a spike. If you add top-down input on top, right, you're going to get a burst instead. So it's like a conversion, right? Change the coin from $1 to $5 or whatever. And the assumption we're going to make is that this is something that happens stochastically. Right? Because there's background noise. So sometimes that works, sometimes it doesn't. And so what that basically means is that if we have somatic input, this input will be represented in something that we call events. Right? It's just a question, is there activity or not? A single spike would clearly be indicative of somatic input. A burst would also be indicative of somatic input because it's, you need the somatic input to trigger activity in the first place. Okay? So if we don't distinguish between spikes and bursts, we might get a handle on what the somatic input was. But what's more crucial is that the dendritic input will, if this is stochastic, just change the proportion of events that actually turn up as bursts. Right? So how many of these events are transformed into bursts is going to be encoded by dendritic input. And so that means we can probably just decode both of these informations from the population of neurons if we distinguish the spike patterns. Okay? So if we think about bursts versus single spikes, both of these signals, both the dendritic and the somatic input, can be decoded. And that's basically the idea that I'll try to sort of unwrap in the following. Okay, so that's just the encoding, if you wish. It's the biophysics of the pyramidal cell that sort of suggests that there's an encoding of these two signals. And I'll later on talk a little bit about decoding by means of biological means. Okay. Good, so let's see. Let's just visualize this very easily, how the code works. So as a function of time, you might have somatic input, that might be rising, and dendritic input that might be going down. You have your population of neurons. They're now very simple neurons. They're just 2 compartment neurons that have this sort of nonlinearity in the dendrite. Happy to talk about the details, but I think it doesn't matter so much here. And so now if you look at the activity that these neurons are going to generate, now let's focus on 3 of them. You might see a burst of activity here and 2 single spikes in these sort of later time bins. Second neuron might have a burst here and a single spike here. Third neuron might just have a burst in the last time bin. Okay. If I wanted to decode from that, what I could do is just calculate something that we call the burst probability, meaning the fraction of neurons that were active and bursted. Okay. So in that time bin, it's just 1 neuron that was active, and that one emitted a burst. So it's 1 burst over 1 active neuron. So this burst probability is 100%. 100% of the neurons that were active actually emitted a burst. Okay. Second time bin, 2 events, 1 burst. So the burst fraction or burst probability is 50%. third one, all three neurons were active, only one of them bursted, so this is 1/3. Okay. And events is even simpler, just one event, 2 events, 3 events, and so that event rate, what we call it, is going up over time. And so now you might see this is something that is, of course, by design, similar to that ramp, right? And this going down is similar to that sort of decline. Okay, so let's make that a little less schematic. This is now a simulation in which we take like a population of neurons. I think it's like, I'm not sure, I think it's 4,000 neurons or so. We nowadays use 4,000 neurons. I think at the time it was 400 or so. It's not a huge number, okay? And so now you have dendritic input that has this weird sort of step-like shape, and somatic input that has this step-like shape. And the way this stimulus is designed is that if you just look at the overall firing rate of that population, it's basically flat. Okay, so you can't really decode the 2 signals from that activity trace, or just from looking at firing rate. But if you look at the burst rate, you see that there's something else going on. Right? So there's something here that is indicative of the dendritic input. And if you take the next step and actually calculate this burst fraction by dividing by the event rate, you see that it now very nicely tracks the dendritic input. Right? And the same thing for the event rate. This is something that very nicely tracks the somatic input. Okay? So it really is possible from that population of neurons to decode both of these signals. And that sort of is just trying to open up the possibility that there's more information in the spike trains of those neurons if you actually take the spike patterns into account than if you just look at the firing rate. And so you're multiplexing both of these signals, and both of them are conveyed at the same time. And I think it's an interesting sort of, there are all sorts of interesting consequences you could think about at that point. Right? You can think about these 2 things being combined post hoc, right? So that you can flexibly design how much you're going to listen to your top-down input and how much to your bottom-up input, because both of them are present. So you just need to, let's say one is the prior and one is the likelihood. Then you could go ahead and, depending on, how noisy your observations are, put more weight on top-down than bottom-up, depending on, the contextual changes in the sensory problem. And so there's all sorts of possibilities that arise at that point. Okay, so that's
so biophysically, Henning, do you think a little bit about which kinds of neural sort of architectures or processes might be susceptible to those different categories as well? So you could imagine, the spiking outputs. Well, anything that has a synaptic connection with a neuron is probably going to notice the firing rate. But to notice the proportion of bursts, you'd probably have to have a relatively long integration, right? You'd have to be able to sort of pay attention, not just what's happened now, but what's happened over the period of time or something like that. And then the burst firing might be something a little bit more like a high pass filter rather than a low pass filter, like being able to notice it. Have a quick refresh time. So I just got hit by a spike, quickly refresh. Oh, I got hit by another one. That's a different kind of a message than if I get hit by a spike, refresh, and then nothing else comes. So you can imagine kind of plausible ways in which the network actually do something with this information too, which is why I think it's really important for us to think about.
That's, if you wish, the next slide, right?
Sorry.
That's if you wish, the decoding problem, right? It's the question of how could like a neural system actually cope with this sort of thing, right? And so one thing first is that long integration times are not really necessary here, because what you can do, because this is a population code, right? So this proportion of bursts is not something that you'd compute over a long time window, but you'd compute it by summing over all these neurons. Right? And so that's something, of course, that is quasi-instantaneous, right? So you can do this almost immediately with maybe the slight holdback that you need the second spike to know that this is actually a burst, right? So that's the delay that you can't avoid. But other than that, this is an extremely fast code. Okay? And the way you decode it is basically through short-term plasticity, right? which is straightforward. So I'll show you in a second. So maybe do I jump? Oh, well, I'll just continue. It's not the next slide. It's like 2 slides down the road. Okay, Mac, please forgive me. Okay, so here, so the idea is that you have this somatic input and you can provide this now as pulses, right? And now what we look at is how that changes the firing rate. And, at that point, you just see the classical gain modulation story that Matthew and a lot of others have observed. If you look at the burst rate as a function of the dendritic input, you see that there's a lot of dependence on different levels of somatic input. Okay, so if you have higher somatic input, you're going to have a lot more bursts. So this is not something, burst rate is not a good measure of dendritic input. But if you look at event rate and burst probability or fraction of bursts, then these lines basically fall on top of each other. So that means you can decode the dendritic input irrespective of what the somatic input was. And here you can decode the somatic input irrespective of what the dendritic input was. So this is in a way, again, like an invariant decoding problem. But that's sort of the two dimensions that work quite well. Okay, so that's possible. Temporal resolution, that's something that I just referred to briefly just now. So what you can do is you can just provide time varying inputs to both the soma and the dendrite, and then afterwards see what happens, right? And look at burst probability, event rate, firing rate, and just plot the coherence between the input and the output as the function of frequency. Right? So it's sort of classical information theory analyses. And what I think what you can tell here is that firing rate is something that really is not an extremely good decoder, right? But the event rate is actually quite good, right? So the event rate basically allows you to decode the somatic input over a pretty broad range of frequencies. And this is 100 hertz, right? So this is a 10 millisecond temporal resolution. So that's, I think, pretty fast. And the same thing is to some extent true for the dendrites. Here you might only go to 50 hertz or so, which is like a 20 millisecond inherent time scale, but that's still comparatively fast. So you don't need to integrate over seconds or so to find out how many bursts there were, because you do that basically across the population dimension. Okay, so decoding. We have an information theorist here somewhere, right? I'm not sure if I should.
Yeah, Joe's here. He'll unmask himself.
Yes, apparently.
Yeah, well, I mean, you can do an information theory of this and try to find out, how much information is conveyed about the two sources of input, right? The soma and the dendrite, and you can do a little bit of math under sort of simple, sort of kind of Poissonish assumptions, right? Maybe I'll skip that, maybe not. So what that allows you to do is to see under which conditions, information transmission is maximized. And there are a few interesting insights here. One is that, that burst probability should be comparatively low, right? So ballpark 1 in 5 spikes, 1 in 3 or 4 spikes should be a burst. Or it should be a burst in order for this to work well. And the second thing you can see is like, what about burst size? Short bursts are the things that are actually most informative, okay? Which is not surprising, because all you need to do is find out whether it was a burst or not. And the second spike tells you everything about that. The third spike is just, a loss of information, if you wish. Okay, and if you do that, you can also try to find out when just a rate code is better than the sorts of multiplexing code in terms of how much information you convey. Right? And so I think what you can do also is plot that as a function of the population size. Right? So how many neurons do you need to average over in order to suppress the noise sufficiently that you get better at decoding those two things? And so basically at the population size of about 10 neurons or so, you're actually better with multiplexing for this specific setting. Okay. So it means you're not going to need 100, like a million neurons or so in order for this to be beneficial in terms of just coding. Okay, decoding finally is the point where you can now think about how you would decode, or how you'd use this stuff, or how you'd use this code downstream in a neural circuit. Now, the idea is that individual spikes, or just events, spikes or bursts, encode one thing, which is the somatic input. So what you should do is take the first spike and ignore everything else, right? And then just count those things from the presynaptic population. And that's something that's comparatively easy to do, because all you need is a synaptic connection to a downstream population that is depressing. Right? That means the second spike should be discarded. And the easiest way to do that is just to depress the synapse such that the second spike basically is not transmitted very well. And this is something that you can see here. So as a function of the somatic input to this population, you can plot the population rate of a downstream population of neurons with depressing synapses, and you see that you also see this level of invariance here. Okay. In the same logic, if you want to detect a burst, then a facilitating synapse is cool. Right? Facilitating synapse in the extreme case would say the first spike is not transmitted, but then you get an accumulation of presynaptic calcium that then allows you to transmit the second spike. Right? And that's something that doesn't work with short-term facilitation alone, but you need basically a second sort of feed-forward inhibition pathway to be combined with that. And that allows you to basically get, again, at the decoding of the dendritic input in a downstream population. So that's sort of the idea, is that, of course, if you think about spike patterns, and specifically on a short time scale, then being sensitive to which pattern you have is something that a synapse can do very well through short-term dynamics. Okay. So yeah, that's basically that postsynaptic part. The other thing that you can mention here, of course, is that this doesn't necessarily need to be a decoding of somatic and dendritic input, because you might wonder, like, why on earth do you encode things so complicated in the first place to only unmix it? But what you can do, of course, is also to just combine these sources of information in pretty much arbitrary ways, just by changing the properties of synapses in the system. Okay, and we know that the form of short-term plasticity of neural connections is highly malleable, because there are things like presynaptic GABA receptors that can switch a synapse from being, basically just standard to very strongly facilitating. And so there's loads of things that could allow you to do flexible integration at that level. Okay, and I think with that, I'm actually at the thank you slide. So sorry for torturing you for such a long time. That's basically an old picture of the lab. We don't have a current one due to COVID. It's very hard to bring people together in close vicinity at the moment. But those are the people who actually did all the work. And I'd like to highlight Joram and Laura, who did all this, this first part of the talk. And the second part was primarily done by Richard Nord, who now runs his own lab. So if you want to see how that story developed, then he's the person to talk to. And he's done beautiful work, also with Blake Richards about what that means for backprop, etc. There's loads of cool stuff that follows from that, and of course people were brave enough to fund me. Thank you very much, everybody, and I'm very much looking forward to your questions.